Hello, this is my chapter 7 9 PowerPoint for biology. Um, we'll get started here. Um, so, the first thing we're going to talk about in today's PowerPoint is viruses and exactly what are viruses consist of, all the different types of things that you need to know about viruses. So, to start with, viruses are everywhere in nature. So, viruses are very abundant. For an example of this would be, ocean water. There's about 10 million viruses per milliliter. And lake water, 25 million viruses per milliliter. So, a milliliter is very, very small, and that gives you a good estimate about the ratio between viruses and you know in water anyways. But viruses can be found anywhere on everything, almost the same as bacteria. Viruses are more, yeah, we'll go with that. Let me get my laser pointer here. But yeah, we're going to keep talking more about viruses and the properties of viruses. So, here's some things about viruses that everybody should know. So, viruses are not considered cells. They are intercellular parasites that infect plants, animals, algae, fungi, bacteria, and protozoa. So, viruses also have a basic protein shell that surrounds nucleic acids. So, think of like a shell, like an egg of protein. Like the shell is the protein, and inside of the egg is your nucleic acids. Think of it like a hard-boiled egg. That's how I think of it, anyways. When it comes to the shell, this is what it made me think of. They can either have DNA or RNA, but they cannot have both DNA and RNA. So we'll talk more about the difference between DNA and RNA viruses. But viruses also lack enzymes for metabolic process. So that's another key thing that we'll talk about in a later slide. But they also lack machinery for making. They also lack for making proteins. It cannot make proteins either. So let's talk about some of the structure of a virus. And I put this picture here from the book. I thought it was really, really good picture that really shows. So like here you have your capsule, your protein capsule, and your nucleic acids. And this is a naked virus, right? And here's your enveloped virus. So you can definitely see the difference between the two. However, as you can see, they both all have these spikes. The spikes are different, but same thing. So viruses are small in comparison to bacteria. So roughly about 2,000 bacteria could fit, or viruses are very small in comparison. 2,000 bacterial viruses could fit in an average bacterial cell. So 2,000 viruses could fit into one bacterial cell. More than 50 million viruses could fit. 50 million viruses could fit in a single human cell. All viruses contain the protein shell that surrounds the nucleic acid. And like I said, I thought this was a real good picture, a good reference on what we're talking about. So we'll move on to the next slide here. So we're going to talk more specifically how viruses are classified. So a few questions that you need to ask yourself when classifying a virus. So, do they infect animal, plant, or bacterial viruses? Is it enveloped or is it a naked virus? Is it a DNA or is it RNA stranded? Is it helix or isosceroidal? Can't really pronounce that word, but I mean, these are some key questions that factor into how viruses are classified, named, and things of that nature. So we'll move on. So we're going to talk about so the next slide here is how do viruses multiply? So there's a few things here about the process of multiplying a virus. So first, the virus will attach itself to a host cell, more specifically the membrane receptors. So a virus cannot live unless it is attached itself to a host. So if a virus is not attached to a host, it mostly lays dormant. But if it does attach itself to a host, I mean, that's the first step of multiplying, is going from that dormant stage to the active stage, I would say. And they mostly do that to the membrane receptors. And the membrane is kind of like, like that's the entrance of cells, So then it enters through the membrane by host range. I mean, that's what it's called. So after it goes through the membrane, it's called host range. That's what it's called, that part. And after entering the cell, a virus will make genomic and proteins to create copies of itself. So what a virus essentially does is it's going to attach itself to the outside layer of the cell, and it's going to use host range, and it's going to penetrate through that cell. And it's either going to attach itself to the nucleus, or it's going to attach itself to the cytoplasm. So that's the key difference between DNA and RNA. So like your DNA strands will only replicate if it has the host nucleus, and RNA will replicate in the cytoplasm. So that's the key differences in RNA and DNA viruses. And after it gets in the cell, like I said, it just makes a genomic and protein. And essentially, that's what a virus consists of. So it just copies and makes more of itself over and over again, either via RNA cytoplasm or DNA nucleus. So, that's that. So, how human health and viruses, how does viruses and human health coincide? So, it's impossible to count the total number of viruses worldwide. However, there has been 268 viruses discovered and named that can infect humans. And another note, it's very rare for a virus to be deadly. So out of the 268, there's not many that are very lethal. However, there definitely are some viruses that are lethal and can cause long-term health problems if infected. And another key note about viruses, antibiotics do not directly affect a virus like it does bacteria. So antibiotics don't work on a virus, but it's still beneficial to take antibiotics, but it's not going to be the same effect as if it was a bacterial infection. So those are some good notes. So we're going to move on to the chapter 9 part. So we got microbe nutrition. So some essential nutrients of microbes are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Nutrition is the process of which nutrients are acquired from the environment and used for metabolism and growth. So this is essentially like we eat food, microbes eat food. It's the same concept. So the two major nutrient types is you got macronutrients and you got micronutrients. And so the micronutrients, these are for metabolism and cell structure, and it mostly comprises of carbon, oxygen, hydrogen. And micronutrients is for enzyme function and protein structure, and it's mostly consists of magnesium, zinc, and nickel. So that's your two major nutrients. So environment factors that affect microbes. So the environment, just like the environment here affects us, environment will affect a virus. So a good example of a factor in the environment that's going to affect a virus or a microbe is temperature. The minimum temperature is the lowest temperature a microbe can withstand. So temperature is a big one. And so the minimum temperature, I mean, that's the lowest point a microbe can withstand, like of it being cold. And so microbes must adapt to environments they cannot influence their own temperature. So microbes cannot change their, like how we fluctuate our temperature. Our body can get hotter or it can make itself colder. Microbes can't do that. So it has a minimum temperature, a maximum temperature, and an optimum temperature. So the maximum temperature is the hottest a microbe can withstand, and the optimum temperature is that Goldilocks zone where it is the fastest rate of growth for the microbe. So every microbe is trying to get to that temperature. So another environment factor that affects microbes is gases. Actually, the gases is around. So oxygen and CO2 are the most affected gases for microbe growth. So those will benefit the microbe the best as being in that type of environment. Microbes will do one of three things. Use and detoxify. Those that can't use oxygen nor detoxify it. Those that do not use oxygen but can detoxify it. So they're going to do one or three things, and those are it. But oxygen and CO2 are the most effective gases for microbe growth. So another little key note is pH microbes. I know this is pretty sad slide. So pH 0 to 1 is called a thermoplasm, and a pH of 7 is called a nitromonoris, and a pH of 12 is a protus. So, you know, obviously microbes have pH levels. Is essentially what I'm trying to say is they have their own pH levels. So, like, obviously, you know, you got your neutral zone, and then you got your acidic zone and your base zones. So microbes are going to fall somewhere in this pH scale depending on where they're from, how they're made, or what they are, you know. So, yeah. So, another thing about microbes is osmotic pressure. So a few microbes called osmophis live in a biofilm with high solute. High concentrations of salt, those are called halophils. So these are microbes that stay in high solutes. They want to be near salt all the time. And obligate halophils inhabit salt. They like to be where the salt is, and they're very salt resistant. Like their membranes are also very salt resistant, and it makes for a good environment for them. And this is where they like to be. So you got all kinds of different microbes, and the rate of growth. So, rate of growth, to complete a fission cycle is called generation or doubling time. The length of generation time is measured of the growth rate or in an organism compared with the growth rates of most living things. The shortest generation times can be 10 to 12 minutes. The longer generation time can be days. But average generation time is 60 to 30 minutes. So, I mean, this is how fast it can grow, the rate of growth, and all of that. But